Thank you for having me. I mean, it's been an absolute treat to be here with all of you this week. This is, I think, definitely the kind of program and workshop that we should be doing more of. So yeah, thank you. So yes, I'm going to talk a little bit about the work that we've been doing, trying to understand the evolution of phenotypic diversity, which is the big overarching question that we try to address in my lab. This is a cichlid embryo. This is a cichlid embryo stained for TBXTA, TBX16, and TBX24. I'll tell you more about them in a bit. No, you can see the eyes there. I think that gastrolux looks very good at generating head structures. But yeah, we've also got some pescoids, which I won't be showing today. Okay. So I'm interested in the evolution of biological form, and I want to understand how it is generated and how it evolves. And over the course of my career so far, I've been very interested in studying how segmented body plans in particular develop and how they can evolve to generate the vast diversity of segmented animals that we see today. Because, in fact, segmented animals make up most of the Earth's animal biomass, with marine arthropods as the most represented group, followed by fish, annelid worms, and terrestrial arthropods, all of which are segmented, of course, which makes a good case for studying the evolution and development of this trait, because if we understand how segmentation is generated and how it evolves, we can understand a high portion of the diversity that we see. Preaching to the choir, but I'll say it anyways. But this is particularly obvious to see or apparent in the vertebrates, where segments which correspond to the vertebrae are known to vary in size, morphology, and type, but perhaps most astonishingly, at least to me, in number, where they range from as few as 10 in some species of frogs to easily several hundred in snakes. And although it's so obvious to see, we know remarkably little about the evolution of vertebral counts themselves. And so in my group, we take an interdisciplinary approach to try to get at this. And today I'm going to talk to you about two levels of explanation that we've been working on over the last couple of years. The first is, trying to understand what is the developmental basis for the high degree of evolvability that we observe in vertebral counts, in the trait that we're after. And the second is, which are the actual developmental changes that lead to the evolution of vertebral counts over short evolutionary timescales. And to do that, we turn to the vertebrate embryo, and in particular to the process of somatogenesis, which has already been introduced in this workshop, but that I'll go over quickly in these next couple of slides. So somatogenesis is the process by which vertebral precursors, known as somites, are patterned in the presemitic mesoderm, or PSM for short. This is another cichlid embryo. It looks a little bit like a chick, I think, in my opinion, more than a zebrafish embryo of the same stage. So interesting changes in geometry already in closely related organisms. So these are the somites, these paired blocks of mesoderm. That's the PSM that Lisa introduced yesterday already, and that we're going to be focusing on today as well. And idealizing this process, during this process, you've got these traveling waves of gene expression that propagate from the posterior to the anterior of the PSM, where they come to specify the somite boundaries as the tissue elongates. And these waves, what they are is that they are an emergent property of a molecular clock that's active in the single cells, that synchronizes with the cells of the neighbors to generate as an emergent property, these traveling waves of gene expression. And conceptually, at least, somatogenesis has to evolve if we evolve vertebral counts, or in order to evolve vertebral counts, conceptually at least, and hold on to that conceptually at least part. And in order to increase vertebral counts, one would expect that either the tempo of somatogenesis, so the frequency of the clock, would need to increase in order to fit more somites in the same amount of time that you allocate to the process, or the mechanisms driving PSM axial elongation would need to change to increase the total duration of the process in order to fit more somites in that way, or of course a combination of the two. And we know that both change in evolution. However, when we started this project, it wasn't clear from the literature whether frequency or duration were most often employed by evolution to increase vertebral counts or to just generally alter the number of vertebrae in an organism, and therefore whether one might be more evolvable than the other. So to explore the relative importance of these two factors, we came up with like 3 idealized scenarios. So in scenario 1, if we look at the literature and we look at the frequency against the number of somites for a bunch of species, if it is the tempo of somatogenesis that changes most readily, we should see a relationship like this with, clock-only evolution, where you're increasing the frequency and you're increasing somite counts. Or alternatively, you are only increasing the duration and not the tempo of the clock. So you'd see a relationship between data points that was looking a bit like this. Or in the third scenario, you would see that the data spread across both axes. So there was no clear relationship between the two. So if we plot the total number of somites formed in the embryo against the rate of somatogenesis, and we just plot it like this, just full-on raw data, we see no clear trend, and we see that the frequency of somatogenesis and the total number of somites is incredibly diverse across the vertebrates. And this would be consistent with something along the lines of scenario 3. But of course, this isn't. And one of the things that I want to point out here in the data set, for example, is that the snapping turtle is the species that makes somites the slowest, with 1 every 19 hours was a dream model system. That is, and the fastest in our data set would be the Mexican cavefish with 1 every 19 minutes or 20 minutes. So really very different. Yes?
So is there some sense in which timing of the age of the animal matters for this correlation if you were to rescale frequency?
Developmental pace matters, right? And we need to take that into account, because obviously if everything is happening more slowly, then you can expect the tempo to also be slower. And when you do that, you do see a correlation between the frequency and the number of somites, pointing out that indeed accelerating the clock has been very important to increase the number of segments. And this was shown already in Olivier's lab here when he did the work on the corn snake. Let me see if I've got the... Yes, so what we're taking is the time to the appearance of the first pharyngeal arches. It's a little bit arbitrary, but it's one of the most conserved features in vertebrates, so that's what we take. And of course, if we do this, you see that the snapping turtle is no longer such an outlier, or as much of an outlier as we see before. And you also see that, in snakes, which we already knew from Olivier's lab, the tempo has been, the increase of the frequency of somatogenesis has been very important to increase the number of somites. And sorry, I use sort of somites and vertebrae a little bit interchangeably, which perhaps I shouldn't, but there we are. So we also see for some predatory fishes, such as the European sea bass here, that they make very few vertebrae despite having elongated their axis. This tells us that, increasing vertebrae is not the, because we tend to associate an increase in the vertebrae to increase the aspect ratio of the animal. That is not the only strategy that they have. They can also elongate the vertebrae themselves afterwards, perhaps selecting for a stiffer body to swim faster. But not all fish do that. For example, we've got here the long spiky head carp, which has also increased its aspect ratio by increasing the number of vertebrae, the number of somites. And in this case, what I think is interesting is that the rate of somatogenesis is almost on par as what we see in snakes. However, they make nowhere near as many vertebrae, suggesting that although we see this very nice correlation between frequency and total Semitic count, in fact, duration also is an important factor in the evolvability of this trait.
Can I ask about whether or not the size of the vertebrae, the length, exactly what you just pointed out, is a separately controlled process? In other words, it happens maybe in the adult, maybe in the juvenile, and not in the embryo?
It does happen a lot later.
Only.
So one thing that I don't think we know is whether the length of the nascent somite has any correlation with the length of the vertebrate down the line. And that's something that at least in our fishes we could look at and we should look at, but I don't think that that's something that's been looked at. But I suspect that those are uncoupled.
So is that similar to what happens in epiphyseal plates in adolescents and animals, wherein there's a very different mechanism associated with lengthening compared to the mechanism associated with segmenting?
I would assume so. And because it happens so much later, and the somites themselves, they themselves grow a lot.
Allometrically now rather than isometrically.
Yes. So I would expect that those to be independent processes. However, we haven't actually established that. I don't think that's been established. Yes.
It seems that one group where this elongated body appeared many times is reptiles. And you don't have that many because the embryos are hard to get.
I don't know why. So these are the species that we were able to find from published literature. So either people have been sort of less inclined, not necessarily because they're harder, but because just people have been less inclined. Sorry. Vodafone. Okay, let me put this in silence. Yes. So I think that is difficult. Yeah, the sociology of science and that. Yeah.
Time we use to scale the frequency?
Yeah. So is the length of time until the appearance of the first pharyngeal arches, which is at the most sort of conserved, the phenotypic stage of vertebrates. It has finished. It has finished. Yes, it has finished. Right. So all of this just to point out that frequency evolves and is a major player, but that duration is also a very important player, perhaps somewhat overlooked in that respect, but still a very major player. And that if we want to understand the evolution of duration of somatogenesis, we then have to start looking at the evolution of the morphogenetic processes that are underlying the elongation of the axis, amongst other things.
These samples?
No, it's sort of implicit, because if they make X number of somites and that's the tempo, then you can calculate the duration implicitly from that, which is why this one has less duration. Yes. Okay. So from looking at this, we started to think, well, somatogenesis must be a very modular developmental process if it can change in these ways to give you all of this evolvability, right? You can evolve the segmentation clock tempo, you can evolve the duration. Perhaps if those two can evolve more or less independently of one another, that really increases the evolvability of this process. It uncouples the two. And there are already a lot of clues around to sort of hint at a decoupling in these two axes, the tempo and also the duration. From the data, for example, the period is not conserved, neither is the duration, but also very importantly, the identity of clock genes is not conserved. So clock genes have been found to be very variable. So you can change the nodes of those networks broadly, except for HER7, and you can change the players, but also the morphogenetic mechanisms of axial elongation are not conserved. And this is something that this crowd knows very well. I'll go through it just very quickly, but in zebrafish, you have this sort of liquid to solid transition in the PSM, which is, and of course I'm going to focus on the elongation of the PSM, although that doesn't happen in isolation of the elongation of the axis more broadly. But of the PSM, you've got, high motility, high degree of cell rearrangements in the posterior, which stops quite abruptly at that jamming transition that they found in Ujjikampas labs, and then the tissue solidifies. This is different to what is seen to happen in what they found in Olivier's lab to happen in chick, where there's more of a random motility gradient along, and then in mouse, we know.
I think there is sort of jamming transition.
Possibly, yeah. Oh, really?
Yeah, what is different is that these cells are more packed in the fish, like we heard that. But I think it jams at some point when.
But it's sort of a continuum between the two in some ways, that you can change the position of the jamming transition and you get from one to the other. Oh, yeah, that's interesting, yeah. And then in mouse, we know a lot less, but we know that there's a lot of proliferation in mouse that, for example, we don't see in zebrafish. So my point with this is just to highlight that the mechanisms underlying the elongation of the PSM do evolve as well, and that those need to evolve in order to evolve the duration of the process, right? Okay. So we set out to study this proposed modularity of somatogenesis, and it was very well established from experimental work that PSM morphogenesis is in fact independent of segmentation clock. You can knock out the clock. Olivier's lab also showed this, and the PSM can still elongate, right? So, all right, we've got independence modularity in one direction already. Can you change morphogenetic mechanisms, although we do see that they change? What's the degree to which you can change them without affecting clock dynamics? And remember that I told you that the cells need to synchronize in order to have those traveling waves of gene expression. So it becomes not immediately trivial to understand, well, to what degree can those cell movements change in order to still maintain the traveling waves which depend on them implicitly, right? So these would be difficult experiments to do. So instead, we take a modeling approach. I've been showing you the simulations all along, and where by taking a model that was published in 2021 by Koichiro Urio in eLife, we are then able to challenge this model with different morphogenetic modes that we find from different vertebrate species. So this model, Koichiro.
Can I just ask, like in that comparative morphogenesis thing you had, it's true for so many tissues, and that they do more or less the same thing but with different modalities. Could the answer be, maybe Lisa can comment on that too, on just the material properties of these tissues are so different to different species that you have to use different modalities just because you're dealing with different stiffness, different risk.
Right, but that's if you look at the extant species. But if you look at the evolutionary trajectories, the fact that they were able to diversify is sort of the question, right? Because that could have been very constrained.
I see.
It could be constrained to the point where you say, this tissue has to grow in a certain way, or else the underlying patterning is going to fall apart, right? But that's not what we see. Yeah. So we were so happy when this model came out. It meant that, they'd done a lot of the very hard work for us, particularly because Koichiro had gone through the painstaking job of calibrating this model to zebrafish data from Andy Oates' lab. So yeah, super grateful to them. And also Koichiro was really instrumental in helping us set up the model when we started working with this in our lab. So fantastic collaborators to have. And what you see here, so I'll just show you the simulation briefly again, or not. Basically, just with words, what you have is that this is representing the cells, is representing the PSM as this sort of, the posterior of it as a half torus, and then it's got these cylinders along the side. And what they have calibrated, or they have calibrated from experimental work, is the degree of mixing along the PSM. But this and then we have Kuramoto oscillators inside each cell that can synchronize with their neighbors. So this model is not a model of elongation, right? This model does not recapitulate the elongation of the PSM. What this model does is that it imposes the cell movements, and then we see what happens to the upstream or to the emergent property of the pattern to see in our case whether any cell behaviors are going to break it, to see in their case how these traveling waves would resynchronize upon DAPT perturbations, right? So this is very different to what Lisa was doing yesterday, where she's trying to understand how those movements actually talk to each other in order to generate length. We don't do that here. We impose the movements to see what happens at a higher level of, I think there was a question, yes?
So you have cell proliferation at the very end, and you'd presumably tell us about what description of movement you use?
So in zebrafish, so hold that thought for 3 slides. Yes? Okay, so just so that I can quickly show some equations, it is a physics and mathy thing. Biology conference at the end of the day. So we've got the segmentation clock, traditional Kuramoto model, where you've got theta of I is the phase of a cell, and how that changes depends on an intrinsic frequency that is an intrinsic frequency of the cell that's different along the posterior to anterior axis to reflect that that's the slowing down that we see in zebrafish. Then a synchronization term with neighbors that are a cell's diameter away, and that's given by a constant kappa. And then also a noise term. These oscillators are noisy. And then they also simulate the position of a cell, which is given by an advection term that also depends on the position along the AP axis, some random motility, some force that determines its position with respect to its neighbors. They can't occupy the same physical space. They don't want to get too far from each other. And then boundary conditions that are imposed from this torus and cylinder.
Have any AP gradient imposed associated with FGF and metabolism and the frequency which changes, for example, in zebrafish? Yes, we have a cell motility. We have a cell motility gradient, which is explicit in the model, but in this expression, implicit in this position here in the advection term, and also in the random motility term. Equation, you also have an omega, which is a function of location.
Yes. Well, sorry, previous.
Which you put in
here. Yes.
So just put in a linear gradient in the phase.
Linear gradient, because in zebrafish they've seen this sort of more yes. Yes.
Sorry, what's the length scale of the local coupling for that?
We consider that it's a cell's diameter. Sort of, yeah. So really touching. Yes, really touching. Sorry.
And the angular velocity in the Kuramoto model change in time as well?
Yes, we don't. Yes, in this case, we don't implement that. So you mean that the velocity of the cell would be affected by?
At each point, doesn't only depend on where you are, but also changes over time when this process starts and when it ends.
Right. So here that asymmetry is given by the position, and the cells are moving from the posterior to the anterior. So you have that reflected to a certain degree. Yes.
At a specific place, the velocity of the cells at those places would not change over time.
No.
No. Yes, exactly. So yes, exactly.
I asked that.
Sorry.
Maybe you mentioned this for intuition, why you get stripes of synchronicity with asynchronicity is that they get too close together, they synchronize their native node.
Thanks for that question. It's so Koichido, before working on this model, works on models in a cube, of cells in a cube. And he has shown very beautifully that you need the mixing in order to then have synchronization in the anterior. That, to me, is not super intuitive, right? But if you want synchronicity here, you need cells to mix, to have mixed. Yes, which is not super intuitive to me, but he has shown this very nicely. Yeah. So I personally, it's not a reflection of the field. I personally do struggle with that intuition. Yeah.
And that happens in the embryo, right? There is mixing.
Yes, very much. There's always more in the species that we've been able to look at so far. There's always more mixing and more cell motility in the posterior. Yes, exactly. Yeah. Okay. So then going back to your question in terms of where the cells ingress, right? So cell ingression varies across species, right? In zebrafish, and by cell ingression, you can understand two things. One, where cells divide, basically, but also literally where cells are ingressing from the dorsal and also from the lateral tissues, for example, during epiboly, when the PSM is still forming, they're ingressing from the side. So in order to start thinking about this, we simulate these three. We always simulate our changes with respect to these three scenarios. One, where cell ingression is random to reflect sort of like the baseline zebrafish post-tail eversion situation where cells are dividing throughout. But then also that dorsoposterior ingression, which you see at earlier stages, and also this dorsoposterior plus lateroventral that you also see at the very early stages, right? To try to understand this. And we see, if we start with this, we see that the clock frequency and the synchrony are robust to the changes in, and this is all departing from parameter values that have been calibrated with experimental data from zebrafish. So yeah, clock synchrony and frequency, super robust to cell ingression position. You can add the cells wherever you want, it doesn't care. It's going to make those, it's going to pass in at the anterior. So to look at synchrony, we look at synchrony at the anterior, which is where it matters, and it doesn't care.
And what about the somat size, these striking width? Does that change as well?
So that is not something that we're able to recapitulate very well, because here, so if you think about the clock and wavefront model, which is probably the easiest framework that we can use to do this, the nascent somite length depends on both the frequency and the speed of the retraction. And so here the speed of the retraction is always the same because it isn't a model of elongation. So that's not something that we can get at with this. Yes.
Yeah, you may want to link also the noise term to the cell division, because I mean there's been some evidence for this.
Yeah.
And whether it's posterior or anterior, you may see different things.
So that I don't know, and that we haven't done.
When you said like the work that they need to mix
Yes.
posteriorly, if you have more noise generated from the division posterior, I mean, I don't know, but that may impact.
So I think that the posterior, so we have also done some simulations on proliferation, and proliferation, Ruth Baker also had previously shown that disrupts the synchrony of the segmentation clock. My sense, and those are in the paper, but they're not in the presentation, but my sense is that proliferation, so if you have proliferation and you sort of stop clock synchrony during that in the synchronization, clock synchronization with the neighbors during that, it's the posterior, it doesn't really matter because you don't have those, but they do disrupt in the anterior. Yes, yeah, we do see that. So frequency and synchrony, robust to cell ingression position. Then we've got these cell motility gradients, again, fit from zebrafish, and my student James decided, I should have said, that this is the work of James in collaboration with Ruth Baker, also at the University of Oxford. And what then James did is that he thought, well, perhaps with this cell motility gradient, oh dear. There we go. So you can see that there's a cell motility gradient. The cells in the back are moving more than in the front. And well, what happens if we change this, right? This is something that we see changes across different vertebrate embryos. And so he explored this, and again, he sees that it's remarkably robust. The frequency is remarkably robust to parameters controlling the cell motility gradient, as is the synchrony. So yellow means perfect synchrony, and here no change to the frequency with respect to what we already have. So very, very robust. We then, the last ones that I'm going to show you are these compaction ones. So in zebrafish, and we've seen in cichlids as well, the axis, the PSM compacts as elongation occurs. So what is is there anything, is this going to affect the synchrony and the frequency? And you guessed it, it does not. And it does not. So at this point, I started off really happy, like, this is great, this is so robust. And then at this point, I was quite worried, like, this is too robust. At some point, like, there's no evolvability if this thing never changes, right? There's just no evolvability. So in the original model, they didn't take delays into account, right? They didn't take delays, and they didn't, they parameterized this kappa, but they didn't explore them already. So already when we start changing the coupling strength, you see that that is a major tuner of the system, tuner of the robustness of the system, as one would expect. So for example, in this scenario, you see that at low kappas, and this is just some other parameter, velocity, maximum velocity of the cells parameter, but you could plot this against other things, and you'd always see that zebrafish parameters are always well within the high synchrony, but as you start to increase the disorder in some way, this starts to creep closer and closer to that boundary, right? So yes, we have that robustness to cell ingression is robust to coupling strength, but very highly dependent on it as well, right? So we have a tunable parameter there.
So are you saying that like you could put a kappa that changes and has like some distribution for each term? Is that what you've done? Or the overall average?
The overall, yes. So we have one for the whole AP axis of the PSM, we have one coupling strength term, which could be location dependent, but it is at the moment. And we've just changed that one to explore how the synchrony at the anterior would change with that. And unsurprisingly, yes, if you lower the coupling strength, it gets to a point where it doesn't couple, right? And where it doesn't synchronize. Okay, good. So what about then those delays? Super stressful day in the lab this was. Because James came back to show that actually, if you change the delay, and this is, if you change kappa, but with the delay that's been measured in zebrafish, which is 21 minutes, already in the random ingression mode, you lose the synchronicity. And of course, here you lose it entirely. So we started to, yes?
What was the coupling delay? I made this.
The delay, no. The delay is 21. The measured delay is 21 minutes. So you can ask, so what they have done is that they have done DAPT experiments to inhibit cell-cell communication. And then they measure how long it takes for the synchrony of the oscillations to come back. So they calculate that there's a delay of 21 minutes.
So the axes are the same as before?
The axes are the same, but I'm just putting the delay, the 21-minute delay that has been measured in zebrafish, right? That was absent from the original paper and from our original simulations.
There is a delay in the phase locking?
Exactly. Absolutely. Exactly. And that already the zebrafish values mess this up throughout, right? So we decided to focus on this one because I should say that now we've switched to a modeling framework called YALA, developed by James Sharp in Barcelona, which does these agent-based simulations like this. But James really had to wait for these results, and this we published just like 1 or 2 years ago, so it's really come a long way. But just for context, we couldn't really do all 3 scenarios. We chose to do the random one, which was the most sort of generalizable one, we thought, to see then what happened. And thank God, yes, at 21 minutes you lose it, but already at 23 minutes you have it back, right? And at 24 minutes you have it back. So what this is telling us is something very interesting. So it matches quite well with the data. It's not exactly 21 minutes, but we recover the synchronicity at 23 minutes. But something that I find very satisfying and very mysterious is that zebrafish parameters seem to be poised at criticality when it comes to the synchronicity and the delays, right? I don't know what that means, but this idea of criticality we already found in the gap gene patterning, and it just seems to follow me around. And I don't know what it means, but there has to be something interesting. There might be something, I don't know. Anyways, I just find it satisfying, thought I'd mention it. And then also, if you turn the delay down, you recover it again. And this is well known from analytical solutions, right? In analytical solutions of these Kuramoto oscillators, you know that you have synchronicity, then you lose it, and then you recover it again, and then you can lose it again when you change the parameters. So I think that what this is telling us is that you can change morphogenesis, you can evolve morphogenesis quite a bit, so long as you find yourself in one of those evolvability manifolds that Sebastian was talking about earlier, right? So if the coupling strength and the delay are playing ball, you can change the underlying morphogenetic modes quite drastically, and thereby evolve somatogenesis and things like vertebral count. Right, so this system, somatogenesis, really has that modular property where, depending on the coupling strength and the delay, you find yourself in these sort of manifolds of evolvability, which is, we propose, why this process is so evolvable and why we see so much evolvability of vertebral counts.
Do you know what sets the delay time, and can you manipulate it in experiments?
So people have, so for example, so this is obviously not signaling between the cells, and if you have more or less ligands, you can control that, right? So one possible avenue of research from this, which we're not going to pursue, but I'm just leaving it out there for people, is to actually look at the amount of these ligands across different species and see whether they are consistently high, consistently poised at criticality, or whether actually they are more variable than we think and we've got this all wrong, for example.
And do you see when you reduce the delay time that you change the phase because you have the boundary?
Yes, we haven't manipulated that. Yes. Yes.
So at least from the previous slide and what you're showing over here, it seems like you have islands of synchrony.
Yes.
The valley in the middle.
Yes.
And if you're thinking about those as where it's more evolvable and where these guys want to sit, how will you cross?
Don't know, but
What's
yeah.
the axis of the plane that might connect these islands?
Yeah, so this is a project for a master student in the future that I'm thinking about recruiting, because if we can speed up these simulations, which we now can, we can perform these evolutionary simulations to explore whether, do you ever need to go from one to the other? Are you sort of locked in one? And if you're locked in one, does that always give you enough evolvability, or perhaps do we see evolutionary/developmental constraints in the evolvability of vertebral counts, such that perhaps that might explain why some classes have a much higher range of vertebral counts than others? Complete speculation, I don't know.
I think I have a related question, which is that, as I understood, these plots are just telling me whether I get synchrony or not for the most part.
Yes, exactly. Whether you can change morphogenesis and still be synchronous.
Right. And I
Yes.
thought, and I think this is what you just said, but I want to check.
Yeah.
A follow-up question is, well, like, you want to be on the manifold where you have synchrony, but you just want to change the pace of it, right? And I get that, like, if you're near this threshold, you're more evolvable in some sense because you could stop getting because my understanding is if you go all this, you stop getting somites.
If you don't have
get somites at all.
If you don't have synchrony, you will miss.
To be in a space where actually there's a slow degree of freedom where you could change the number of vertebrates, but not actually get into this regime where you don't have somites anymore at all.
Right, exactly. So the abstraction here is that if you're within these, you can change morphogenesis to change the duration of somatogenesis. The work that we've done assumes a constant unchanging frequency. So it doesn't tell us anything about frequency changes and how those come about. It tells us something about how you can change the duration of somatogenesis without messing up the synchrony. Yeah, so within those, you can evolve morphogenesis to change the duration. Yes.
So what? My impression is that this somehow can explain how you can maintain robustness while accelerating the thought.
No, the clock is unchanging in our simulations. What this tells us is that you can evolve the underlying morphogenesis very easily to increase or decrease the duration of somatogenesis within these pockets.
Because you make the assumption that cells will desynchronize if the period of somatogenesis is longer.
No, we are agnostic of what. So we are not addressing what would happen if we change the period. So we're testing the modularity from changes in morphogenesis to how that affects synchrony. So we're showing that morphogenesis can evolve within these pockets quite unconstrained and independently without breaking the synchrony.
But when you say evolve, morphogenesis evolves, what do you mean?
I change the degree of cell motility, I can change the degree of proliferation, I can change the position of ingression of the cells, I can change the length of the PSM, which we also show in the paper, but I'm not showing it here, the compaction. I can change those, they're not physical properties, they're almost structural properties perhaps of the PSM without breaking synchrony, which wasn't obvious when we started because obviously synchrony emerges from cell-cell communication.
I guess we need the main parameter. I mean, there are two ways you can play with the system. One is with the ingression and the other one is with the period, the frequency.
Right. Right. So we're looking at what things like ingression would do. Yes.
So your coupling coefficient there, what is the physical variable that I could intuitively relate it to? So could it relate to density, for example?
Kappa?
Yeah.
Kappa is taken to mean notch delta signaling between the cells.
So it would be proportional to density, right? How much contact I make.
Exactly. Or how many ligands on the cell membrane.
Will this model also explain the end of,
No. No. How long it lasts. No.
Because you lose synchronicity if you get rid of density.
Yeah.
If you reduce the density, if we run out of cells.
Yeah. We didn't do those simulations. Actually, perhaps we should. I don't think, perhaps we should. Yeah. Like how small. Yeah.
I wanted to ask related to that. You said proliferation. Is it the rate of cell division?
Yes.
Okay, it is the
Yes.
rate.
Yes. So how much of it? And that's a limiting factor too.
Yeah.
That's a limiting factor too. You can't just proliferate. Yes.
Use new cells into the system. Like, what's the initial phase that they take?
I think yes. So ingressing cells don't have any. I'd have to double check, but I think that we assume that they aren't expressing whatever clock gene or phase of 0. I think. Sometimes they're ingressing from sort of like the surrounding, and so then it's through the coupling. Yes.
Or explain the evolvability of cellular mechanisms, but are the endpoints the same, like the total length of the process of somatogenesis and the number of somates, or are we?
Right.
So I think that the implication is that if these processes can change, then so can the duration, right? So I'm not showing that, but that's the implication. If these processes can change, so can the duration. Okay, thank you. So in the next 15 minutes or so, I'm just going to switch gears a little bit and show you some, and just to sort of round up, we think that this supports the idea that somatogenesis is a modular developmental process and that axial elongation and the segmentation clock can evolve independently, thereby increasing drastically the evolvability of traits derived from it, such as vertebral counts. Okay, so I'm going to switch gears now. But this has been sort of a conceptual exercise to try to think about, what is it about the developmental process, patterning somates, that is evolvable and that can therefore give rise to to the evolution of different vertebral counts. But now we wanted to see what happens in practice, right? And to do that, you need to look at what the developmental changes are, what the developmental basis is that leads to the evolution of vertebral counts, but over short evolutionary timescales. We've been very good as a field in studying somatogenesis and the general principles underlying somatogenesis, looking at models that were convenient for developmental biology, right? Zebrafish, mouse, chick. If you want to extend this to arthropods, right? So, but if we want to know what changes actually give rise to increased vertebral counts, you have to look for a different set of species. And I'd been looking for that different set of species for quite a while. And I decided that the ideal system in which to study the evolution of segmentation or of somatogenesis would be a group of species that were phenotypically diverse, while closely related, genetically conserved, preferably, although that's a tall order, as well as experimentally tractable. So these were things. And it was like one of the best days of my professional life when I came across these guys, Lake Malawi cichlid fishes, because as you will see, they really tick all the boxes. So in the Lake Malawi radiation, there are more than 850 species with vertebral count diversity of between 28 and 42 vertebrae. They are very closely related, having all evolved less than 1 million years ago from one single common ancestor that is still sympatrically evolving in the lake. So you actually have the common ancestor still there and in the lab. They are genetically conserved with a genetic divergence across species on the same scale as within human populations. So with the genetic diversity that we have used to do nothing, like taller, shorter, curly hair, blonde, they have evolved 850 species and changed their body plan to change the number of vertebrae, which is insane, right? Together with a lot of other traits. And of course, they are experimentally tractable. People love them to do genomics. So then all of these species are sequenced, very, very convenient for us. And you can do CRISPR. We've established live imaging recently, HCR, single cell tracking, and things like that. So just a little bit about them. This is a female with her brood, and one of the fun things about Lake Malawi cichlids is that, boom, they have amazing maternal parental care, sometimes the fathers, often the mothers, and she will keep the embryos in her mouth. She won't eat for 3 weeks whilst they develop, and then, like all of us, she'll try to encourage them towards independence, and they will resist it for a while, right? So we have them in the lab. We have a bunch of species in the lab. I just wanted to point out for Maha's sake that the egg sizes vary. I'm interested to see whether the sort of curvature and that kind of stuff varies, but we have found no correlation between the size of the egg and the vertebral count, just to anticipate perhaps that question. And they are different to zebrafish in many ways in terms of their development. One of the ways in which they're different is that they start elongating their axis and undergoing somatogenesis whilst they're still undergoing epibole, right? So because the egg is so massive, comparably, the embryo is on the scale of a zebrafish embryo, but the egg is so massive. I think this is a centimeter. And it's like a chick. It's basically a chicken fish. Yes. It's basically a chicken fish. So, and that's your embryo there, which looks a lot like a chicken embryo to me. And that's just in with some dextrin. And so here is the leading edge of epibole, right below the axis. And it won't be until a lot later that you actually do blastopore closure as such. And that's at the phenotypic stage, way after somatogenesis is completed. And we have a movie of this, which I wanted to show Lisa. Because this is that leading edge of epibole, right? So these are embryonic cells as well, but they are extra-axial embryonic cells. And I don't know, but perhaps some force, what's generating length from the inside versus what's happening around it. Perhaps is also mechanically involved in the elongation of the axis in this species. I don't know, I'm very tempted. I have a light sheet now that I'm going to learn how to use in the next couple of months, and that's the first thing I'm going to chop. And we can do a little bit of single cell tracking, and we can do some nice HCRs as well and quantify. And I'm going to just rush through this, but I'll tell you more. One of the things that I want, so it's one of 3 radiations in Africa. It's the one that's, so Lake Victoria is much younger, 100,000 years, and Lake Tanganyika is older, around 10 million years old. And then you've got the rivers, which are even older, cichlids in the rivers that are even older. So it's a dream evolutionary experiment in the sense that what I'd like to do next is take species that have increased their vertebral counts in these other two and see whether they follow the same kinds of trajectories. Just very quickly, we have looked at, so this is almost that, so I won't go into it very much, but I want to just show you this data set that we've collected of more than 4,500 specimens, almost 600 species across the African radiation, and we've counted vertebral counts, anterior, sort of precaudal and caudal ones, and we have this data set of whole body micro CT scans. It's annoying because people, when they do CT scans of cichlids, they just do the head. So no one's that bothered with posterior structures. And for those of you interested in morphometrics, there are a couple of talks during the week. This is the pharyngeal jaw, which is fused in cichlids, and it's very, very specialized depending on what these fish eat. So this one is, I think, a snail crusher. And you can see from that dentition that it's really evolved to crush snail shells. But just very quickly, I'll skip through a lot, but we've done some modeling as well. And so what I'm showing you here is that this is Lake Malawi, and vertebral counts have increased. So they have increased. These fish have evolved increased vertebral counts various times convergently, right? So this is the common ancestor of the radiation who's still around. This is a very early branching, 400,000 years ago, branching species, and they all have evolved increased vertebral counts. But you also see that they're starting to evolve increased vertebral counts here and also over here. So we've got this species in the lab, one of them, and this guy, these guys here, and a few others. And we can do some embryology, right? So we can start to see, well, which are the changes that actually underlie at these close evolutionary timescales the evolution of vertebral counts. So the work that I'm going to show you is on Astatotilapia calyptera, the common ancestor of the radiation, which make 30 vertebrae, and Rhamphochromys chilingali, which make 38. These fish are big, by the way. Like these guys are like this, and you have to extract the eggs, and it's involved, but it's good.
Can we do transplant experiments between different vertebrate fishes at early embryonic stage?
So transplants on the egg, I think, are going to be very challenging. We have started using explants and gastroloid models. And I think that in those, if you're lucky enough that they breed at the same time or at the time that you need them to, those should be more durable. Yes, but not, we haven't done those yet. And so when we start comparing somatogenesis between these two species, my money was on the segmentation clock, right? Because I started this 5 years ago, and it was when Miki Ebisuya was looking at all of those differences, and also James Briscoe published the dynamics of neural differentiation things. So my money was on it's not. The rate of somatogenesis is very, very similar between the two. It's a little bit faster in chilingali, but it's very similar between the two. And so if you calculate this, you need 8 more hours of somatogenesis to make the extrasomites that this guy has. So it's a difference in the duration of somatogenesis. So what might it be that underlies those differences in duration? Yeah, per hour, yes. No. So this is the frequency. So that would be 1 somite every slightly less than an hour, and then 1 somite every hour. Yeah. So we started doing, so on fixed samples, this was before we could live image them, just looking at how morphogenesis, to try to infer how morphogenesis might have differed between the two. And you already see that, at mid and at late stages, they look very similar. But at early stages, you already start to see differences in size. And you see that the Rhamphochromys embryo is larger than the Calyptera embryo at the same stage. And so my student Shannon started to quantify this, and she saw that the embryo proper is a bit longer in Chilingali than in a Calyptera, that the PSM is a little bit, but that the PSM, the biggest difference is that the PSM is initially much larger disproportionately much larger in Rhamphochromys than in Eucalyptera. You can see this by eye already at 9 somite stage versus 10 somite stage. And when we quantify this, we see that the Rhamphochromys PSM is larger in every dimension that we measure. So it's larger in length, 1.2-fold difference, and it's larger, much larger in volume, 1.75-fold change in volume, and it has more cells, 1.57-fold difference in cells. And if we use all of our, measurements that we've taken, we see that of the PSM and the unsegmented tailbud, we see that these only ever segregate by species at early stages, but that at mid and later stages, they are the same in PCA space, right? Which again is one of those ways that biology has to just not care about what you thought was going to happen, right? Because we've done all of this work conceptually thinking about how somatogenesis is evolvable. Turns out I don't think it's changed. I think it's the initial conditions that have changed. It's the initial size of the PSM, possibly with the same underlying morphogenetic mechanisms and the exact same tempo that leads to 8 more somates for free. Right? So you don't need to evolve somatogenesis in order to evolve vertebral counts if you evolve the initial conditions.
So can you generalize this correlation between the PSM size and the somatogenesis duration beyond these two species? And also, can it be? Do you need to scale it? Because like the embryo size may differ.
So I've shown that embryo length is a little bit larger in Rhamphochromys than in Calyptera, but not that much. So the PSM is disproportionately larger in this species than in Calyptera at the onset of segmentation. And to answer your second question, I don't know, I'll come to this in 2 slides, but I don't know whether we can generalize. I'll give you reasons for that in a second.
But was it wider too?
Yes. It's wider.
The whole axis was wider. Does that mean anything? If you go to those pictures where you show the PSM was bigger.
This one here? Or
No,
the quantifications?
The quantification.
Yeah. So that's length.
You had pictures.
Right.
Yeah, this one.
Yeah. We also see that it's wider. So I've shown you a subset of the.
Isn't that mediolateral axis? What are those cells? No, a bit anterior next to the
So that would
the yellow ones above and below the red.
All of these. So that's the PSM.
The whole thing is the PSM.
The whole thing is the PSM, and so that would be different different cross sections. Yes.
Just a follow-up question, but what about the size of the soma?
Yes. So they differ a bit. We have this in the manuscript. In fact, which is interesting, they are a little bit larger in Eucalyptera, which is the one that makes less somites, which would be consistent with a slower clock and the same speed of retraction. Yes. So, yeah. Yes.
So the PSM is larger. Does that mean the length and FGF levels are higher, or is it like the big front is?
We haven't. So I think that this is specified during gastrulation. So one thing that's dissatisfying about this is that we're sort of kicking the can up the road a little bit, because I don't know from this work what specifies the PSM. So we're starting now to generate transgenic lines, mesogenin reporters, and other PSM markers to try to see what it is during gastrulation that leads to this difference in the proportions that are allocated to the PSM specifically. Is it because there's just more mesoderm in one species than in the other? I don't know, but this is before we care. These are initial conditions. This is before we care about FGF and Wnt. Yes.
You said it, but I couldn't do the math in my head. You said there were more cells and more volume. Is there a change in density
Yeah, we have measured this, and there is no change in density along the AP axis, like in zebrafish. There is compaction over time, like in zebrafish, but there is no difference across species.
Okay, okay. And then the follow-up question, of course, is motility.
So that's the live imaging is going to help with that, because all of this at the moment we don't have. So all of this supports the sort of more parsimonious explanation that morphogenesis hasn't changed, but until I have the video, I can't really say.
Yeah.
Yes, yes, yes.
You know, I like it that you're saying initial condition is playing a role, but I'm stuck with the end.
Yes.
So why do you stop at 8 and not at 50?
Yes.
Yeah, right?
So this is a long-standing question in the field, right? So we know, for example, that Hox gene expression signals to the end of somatogenesis. So it might not be a process intrinsic decision to make, right? It might not be a process intrinsic. But then also, how does the Hox code talk to somatogenesis and how that evolves over these short evolutionary timescales? I don't know. The proportions also change, right? The number of abdominal rib-bearing vertebrae versus caudal vertebrae also change.
And the PSM size remains the same over time as
No. So yeah, the PSM, if you look at it, it's shrinking. But if you look at it, taking into consideration the length of the nascent somite, it is actually elongating at the expense of height and width, right? So you have to generate length in order to fit you have to rearrange the tissue to generate length in order to fit the species-specific number of somites. But if you look at it, it's just getting shorter. Yes. So now I'll just wrap up, but I'll leave you with sort of that sort of idea of generality that was raised a minute ago. So if we think about like Waddington's landscape, you know, Waddington gave us a view of the underbelly of his landscape with these ropes and pegs, suggesting that either during development or over the course of evolution, these could be used to change the surface of the landscape. And now this has just gotten me thinking that actually it is the initial conditions that are easier to change, right? And that might be also quite a direct readout of the environment, complete speculation, quite a direct readout of the environment using, for example, things like maternal factors, which is something that we're very interested in the future, particularly in this case, considering that the actual genomes of these two species are practically the same. So either something very specific has changed in the genomes or nothing has changed at all, and it's I don't want to say plasticity.
It's fed the egg already.
Mom.
Yeah, the mom.
But
Mom
to be fair, all of my bets have been wrong throughout this process, as you have seen. So I don't know. Yes.
What would be the situation like this, for instance, if you change slightly the expression of genes and activate slightly prematurely some more posterior genes and delay regression, and hence you get this kind of phenotyping.
Yeah.
that could appear.
I think Hox genes, if you change very slightly the timing of Hox genes at the time when you have, the production of these cells, then you will change the number of cells produced, and hence you will see this kind of phenotype.
So one thing that we're doing at the moment is generating surrogates between species, which is when you sterilize, render one adult sterile, and you inject the PGCs of another species. And you can do this in fish. It's mental. And then if you breed surrogates, then the zygotic genome is from one species, but presumably a lot of the maternal components will be of the host species. So I think that maybe that's there. But then last slide, I promise, going back to that question of generality, well, I don't know how general this is. Is it perhaps because I'm looking at something that happens so close to the phylotypic stage that we know is so constrained? We don't know why it's so constrained, but we know that it's so constrained. And so therefore, if you want to change vertebral counts, it's actually easier to change the initial conditions of the process than the process itself. Do you think it's general? Yes.
Just they can reuse at exactly the same right number of single RNA sequencing.
Yes, there is money for that at the moment. I can't decide, maybe we should chat about this because I can't decide on the stage. So at early stages, I guess. But if it's already different, that won't tell me how it's become different. So I'm struggling with choosing the stage for that. So yeah, maybe you can help me think about that.
Yeah. Sorry, I'm having a difficulty understanding the causal relationship between the lengths of the PSM and the durations of somatogenesis.
Right.
Because I've been thinking the lengths of the somatogenesis would rely on how long you can maintain the NNP population in the tailbone. But then But then I also noticed that when you measure the length of the PSM, I saw the long period of the plateau, and then it starts to decline. But then wouldn't it be also possible that you can manage to change the duration of the plateau, not solely based on like consuming the length of the PSM?
Okay, so perhaps the second question, we need to have a chat over lunch, and that will be a better way of going about it. The first question, NMPs. NMPs in zebrafish, here we haven't looked at them because our SOX2 probe doesn't work reliably. But at least in zebrafish, they've shown in the lab of Ben Steventon in Cambridge that you don't really have NMPs per se. In the tailbone of the zebrafish, the population is very small, and it's not actually playing a role substantially towards the elongation of the axis, right? So you have NMPs early on during gastrulation, as one would expect, but the cells in the tip of the tailbone in zebrafish that are co-expressing SOX2 and brachyury never have progeny that do both, right? So they're not NMPs per se, and there's very few of them, and they're not contributing so much. All right, so I have to thank the wonderful, wonderful people who have done the work. So James did all the modeling as well as measuring the rates. Shannon has done the experimental embryology. Callum, I wasn't able to show you much of his work, but he's done the macroevolutionary work and the CT scanning work as well. And Georgina, who was actually in Santa Barbara before in the lab of Uche Campas, was an angel from heaven that came to my lab because she was a postdoc and she wasn't sure what to do. And she came back to the UK and she joined us as an RA, and it just changed everything. So yeah, with that, I'll thank collaborators, funding sources, and all of you for your attention.